Module 3, Principles of DNA Sequencing. Learning objectives. The learning objectives for this module are to explain the principles of DNA sequencing methodologies and analyze sequence data. Principles of DNA sequencing. DNA sequencing is the ultimate DNA test. Until recently, Sanger or dideoxy-DNA sequencing was the predominant method. This method relies on amplifying individual DNA sequences. Each amplified DNA, nested sets of labeled DNA copies are made and then separated according to size using gel electrophoresis. It's one of the earliest methods of DNA sequencing and was used for many, many years. Although it does have a limitation in that it is a single target directed type of DNA sequencing. So one cannot in parallel sequence different types of DNA simultaneously. It also relies on electrophoresis, either in slab gel or capillary gel, and it does involve essentially DNA synthesis in a test tube, and these different DNA synthesized fragments are separated according to size using electrophoresis. In the last few years, completely different technologies have allowed massively parallel DNA sequencing. These methods do not involve electrophoresis and allow us to probe multiple targets simultaneously. No attempt is made to obtain the DNA sequence of just a purified DNA component. Instead, millions of DNA fragments are simultaneously sequenced without the need for gel electrophoresis. Let's consider dideoxy DNA sequencing in more detail. Shown on this slide is a dideoxynucleotide. This molecule forms the basis of the dideoxy sequencing method. Examine the structure of this molecule in closer detail. Shown here is the generalized structure of a 2', 3' dideoxynucleotide triphosphate. The sugar is a dideoxyribose because the hydroxyl groups attached to both carbons 2' and 3' of ribose are each replaced by a hydrogen atom, shown by shading. In a traditional dideoxynucleotide, the 3' hydroxyl carbon would have a hydroxyl group attached to it, which is critical for continued extension of an addition of a dideoxynucleotide by the DNA polymerase. This slide exemplifies the premise followed in dideoxy DNA sequencing. In dideoxy DNA sequencing reactions, a DNA polymerase uses an oligonucleotide primer to make complementary sequences from a purified single-stranded starting DNA template. The sequencing reactions include dideoxynucleotides, which compete with the standard deoxynucleotides to insert a chain-terminating dideoxynucleotide. Different labeling systems can be used, but it's convenient to use labeled dideoxynucleotides that have different fluorescent groups according to the type of base, as shown here. So that would mean 1-fluorophore for dideoxy A, 1-fluorophore for dideoxy T, 1-fluorophore for dideoxy C, and 1-fluorophore for dideoxy G. The DNA copies will have a common 5' end defined by the sequencing primer, but variable 3' ends depending on where a labeled dideoxynucleotide has been inserted, thereby producing a nested set of DNA fragments that differ by a single nucleotide in length. A series of nested fragments that differ incrementally by one nucleotide from their common 5' end are then fractionated according to size by gel electrophoresis. The fluorescent signals are then recorded and interpreted to produce a linear base pair sequence, such as the example given in this slide. This slide depicts the process of slab gel electrophoresis. We will discuss slab gel electrophoresis in the context of that of dideoxy DNA sequencing. In slab gel electrophoresis, which is typically conducted vertically, individual samples are loaded into pre-formed wells at one end of a solid slab of polyacrylamide gel. The samples migrate in parallel lanes towards a positive electrode. Typically, there is one lane for G, one lane for A, one lane for T, and one lane for C for a given template. The separated nucleic acids can be detected in a variety of ways. In one method, after the end of electrophoresis, the gels can be stained with chemicals such as ethidium bromide or cyber green that bind to nucleic acids and fluoresce when exposed to ultraviolet radiation. Alternatively, the nucleic acids are labeled with fluorophores or radioactivity before electrophoresis, and during or after electrophoresis, the individual nucleic acid fragments are detected either using a fluorescent detector or autoradiography with X-ray film. This slide describes capillary gel electrophoresis. The disadvantage of slab gel electrophoresis is that it is very labor-intensive and technically demanding. The modern trend is to use capillary electrophoresis, which is a largely automated process. Fluorescently labeled DNA samples migrate through individual, long, and very thin tubes containing polyacrylamide gel. A recorder detects fluorescent emissions as samples pass a fixed point. Modern dideoxy DNA sequencing uses capillary gel electrophoresis, as do many different types of diagnostic DNA screening methods that will be discussed later in this course. Commercially available DNA sequencing technologies. In the early mid 2000s, new sequencing by synthesis methods were developed that could record the DNA sequence while the DNA strand was being synthesized. That is, the sequencing method was able to monitor the incorporation of each nucleotide in the growing DNA chain and to identify which nucleotide was being incorporated at each step. The new sequencing technologies, often referred to as next-generation sequencing, or NGS, represented a radical step change in sequencing technology. Standard DNA dideoxy sequencing is a highly targeted method requiring the purification of specific sequences of interest that are then selected to be sequenced one after another. By contrast, massively parallel DNA sequencing is indiscriminate. All of the DNA fragments in a complex starting DNA sample can be simultaneously sequenced without any need for gel electrophoresis. The difference in sequencing output is therefore vast. Table 3.3 lists various next-generation sequencing technologies that are commercially well established. Some of these require amplification of the starting DNA, while others rely on unamplified starting DNA, single molecule DNA sequencing. They vary in different parameters, such as read lengths, the length of DNA sequence generated per starting DNA, run lengths, and the number of different DNA sequences that can be conducted in parallel. By comparison with the standard Sanger dideoxy sequencing, the next-gen sequencing methods generally have high intrinsic error rates in base calling, but the final reported sequences are much more accurate than the initial reads after quality filtering and comparison of multiple read sequences. And, importantly, they have significantly cheaper running costs per base